In this objective, we describe the three types of responses that cells make to signaling molecules. There are three types of cellular responses. So either the ion channels are open or closed in response to binding to a neurotransmitters. Um, as we saw in our previous objectives, when neurotransmitters such as acetylcholine bind to the ion or ligand-gated channels, there is an influx of positively charged sodium ions, and this stimulates um, an electrical response in the cell. Whereas when the cells are bound, or when the ion channels are bound by GABA, this stimulates influx of negatively charged ions, and it'll inhibit any response. So there will be no production of any electrical impulse. Now, when the ion channels are open, it depends on the type of charge that enters the cell. That's going to determine whether or not you get a response. But when the ion channels are closed, no ions are moving through the channels. So therefore, this could either prevent or allow for a response to occur. So again, it depends on the charges that are entering the cell when the channels are open, and that's going to determine the response. Okay, another type of cellular response is enzymatic activity is altered. So the example is receptors on white blood cells. So when bacteria infect the body, white blood cells have surface receptors that are going to detect the bacterial proteins. And so these proteins can bind to the white blood cell, and then this could cause enzymatic activity within the white blood cell to change, in that it will impact how the white blood cell will move. So, for example, the white blood cell will want to move toward the bacteria so it can destroy the invading bacteria. So, enzymatic activity is altered in that white blood cell. In terms of specific gene activity that may be turned on or off. So one example is the steroid hormone that can regulate gene expression. And so these steroid hormones can, or the receptors for the steroid hormones can bind to, no, I said that backwards. So the steroid hormones have receptors in the nucleus, all right? So these nuclear receptors, when bound, can regulate gene expression or gene activity. Another example of where gene activity can be regulated is through the RAS pathway. Now, although this is another pathway that you guys are seeing again, you don't have to know all the details for this pathway. The main thing is that in this particular pathway, tyrosine kinase activity or tyrosine kinase is bound by a growth factor. So this is going to signal dimerization and phosphorylation, and this will activate a G protein. The G protein is known as RAS. This is a small G protein. This particular protein has enzymatic activity and can go on and trigger a cascade of signaling events to other kinases, all right? So it's going to communicate with other pathways. It's going to communicate with the MAP kinase pathway. So we know kinases phosphorylate other things. And this will eventually result in gene activity being altered, okay? So the genes that are altered in this particular pathway includes genes that are responsible for cell division, cell growth, cell differentiation. And we'll talk about cell differentiation in later chapters. But for now, note that the activity of the genes that are altered in this particular pathway are responsible for cell division, okay? So the example written on your slide is that mitogen induces mitosis. So mitosis is a form of cell division, okay? So that is another way that gene activity can be altered. It's through the RAS pathway and also through the steroid hormones or those intracellular receptors, okay, that bind to the DNA. In this learning objective, we will compare or contrast signal amplification with signal termination. With signal amplification, a single signaling molecule can lead to changes in millions of molecules at the end of the signaling cascade. So as it relates to signal amplification, some signaling molecules can bind G protein coupled receptors and activate many G proteins, okay? That could lead to the activation of many G proteins. And so these G proteins will go on and bind adenocyclase, and then that can in turn activate many um, cyclic AMPs or production of many cyclic AMPs. So we know that adenocyclase converts ATP to cyclic AMP. So when you have many of these molecules activated, this will go on to activate more protein kinase molecules downstream or after activating the second messenger. And so therefore you're amplifying the signal. Now, with many of these signals, they must be terminated, okay? So we have to return the receptor to its inactive state. And so how do we do this? So one of the ways to return the receptor to the inactive state is to turn off our second messenger, all right? So to convert it to an inactive form. So we have adenosine triphosphate here. Okay, so you see the 3 phosphate groups, the sugar and the adenine, all right? So again, cyclic adenocyclase is responsible for converting ATP to cyclic AMP. Cyclic AMP, in order for the signal to be terminated, or any downstream effectors to be terminated, we have to convert cyclic AMP to an inactive form. So phosphodiesterase is the enzyme that will regulate cyclic AMP activity. So it turns off cyclic AMP by converting it to AMP or adenosine monophosphate. So notice the 1 phosphate group, the adenine ring, and the ribose here. And in our final objective for this chapter, we cite evidence for supporting a long evolutionary history for cell signaling molecules. So some signal transduction pathways are found in diverse organisms are quite similar. So G proteins, protein kinases, and phosphatases have been shown to be highly conserved across organisms, and they are a part of signaling pathways in most organisms. And because of the similarities in many species, this would suggest that there is an evolutionary relationship. Cell communication first evolved in unicellular organisms. So your choanoflagellates, these are unicellular protists. And they are thought to be the most recent ancestors of animals. So they're used as models for studying early evolution. Now, protein kinases, G protein linked receptors, they've all been identified in choanoflagellates. Because of this, these proteins have been found in these unicellular organisms. This would suggest that these proteins are necessary for cell communication, and it would also suggest that these proteins have evolved before the evolution of animals.